Okay, today's speaker is Rayna Bell. She's a new postdoc in Jim McGuire's lab, and she comes to us from Cornell University. She's one of two president's postdocs in the MVZ, and that were awarded this year. Andreas Chavez is the other. System-wide, so president's postdocs are awarded across all UC campuses, and I think system-wide there were something like 15 or 20 of these awarded. And two of them are in the MVZ, so that speaks very highly of these individuals and in the MVZ. She graduated from Cornell just recently, and she is fascinated by the African reed frog and is dedicated to exploring diversification and the evolution of sexual dichromatism in this group. And apparently you find that making discoveries about poorly studied organisms is extremely rewarding, and part of the reason you've dedicated your life to biology. So today we're going to hear about comparative population genomics of island endemic reedfrogs. So welcome, Rayna.
Okay. Thanks everyone for coming. So I just got here a month ago, and so I'm going to talk about the work that I did, or some of the work that I did at Cornell. And then I'll spend a little bit of time at the end talking about some of the things that I'm planning to do in my next 2 years here at the MVZ. So, oceanic islands accumulate endemic species via two key mechanisms. Dispersal from continents or adjacent islands, and then subsequent divergence from those source populations, or diversification in situ of resident species. And the relative importance of these two mechanisms, in situ diversification and dispersal, they vary predictably with a number of island characteristics. So the relative contribution of in situ diversification increases in islands that are older, larger, and more remote. And then dispersal dominates in archipelagos that are comprised of numerous small and young islands that are near continental sources. And there are hundreds of studies that quantify the relative importance of these two mechanisms in generating diversity in oceanic islands. But many of the classic systems are focusing on archipelagos that are young and very remote, like the Hawaiian and Galapagos archipelagos, or focal species that are really good dispersers across saltwater barriers, like birds. And of course, groups that have diversified extraordinarily within archipelagos, so Bengal would be a good example of this. And the system that I'm going to talk about today differs from these more classic study systems in that the islands are pretty old, and they host a number of species that we would tend to think of as being poor dispersers across saltwater barriers. So things like shrews and fossorial reptiles and amphibians. And a lot of these groups haven't actually diversified very much within the archipelago. So the Gulf of Guinea archipelago is located on the Cameroon volcanic line here off the coast of West Central Africa. And the oceanic islands have remained isolated from the continent throughout their existence. But because they're relatively old, they've accumulated a number of endemic species. And like I mentioned, some of these endemics are from groups that we tend to think of as being absent from oceanic islands, so amphibians and fossorial reptiles. And this extraordinary diversity is actually composed of taxonomically disparate groups. So this suggests that a lot of the diversity on these islands arose from dispersal from the continent as opposed to diversification within the archipelago. And we also see a number of sister species across taxonomic groups that are shared between islands in the archipelago, suggesting that dispersal within the system has also been an important source of diversity. So the islands host 7 endemic amphibians, including a caecilian, which is pretty remarkable for oceanic islands. And the prevailing hypothesis for how these amphibians would have colonized these islands is via assisted dispersal on vegetation rafts. And these large vegetation rafts are swept down major river drainages following big rain events. And there are 3 major rivers that empty into the Gulf of Guinea that could potentially serve as sources for these types of vegetation rafts. So the Niger River, which if you just want to pretend that the map extends, originates up here in West Africa, and the Congo River, which originates all the way out in East Africa, and then the Ogooué River, which is just a smaller river but originates here in western Central Africa. And even though the Ogooué River is the most proximate to the islands, it's only about 250 kilometers away, the currents in the Gulf of Guinea direct freshwater plumes from the mouths of these two other rivers towards the islands. And so this means that vegetation rafts that are originating all the way out in East Central Africa or in West Africa could feasibly reach the islands. And so we can try to reconstruct the colonization history of the island endemic amphibians based on the geographic distributions of mainland species that are most closely related to the island endemics. And there are a number of phylogenetic studies that include samples from the islands and mainland congeners that have attempted to do this. And they tend to identify, and there are studies for 6 of the 7 amphibians, they identify mainland putatively ancestral or sister species that are found in East Africa. And this is somewhat surprising, but the Congo River does originate all the way out in East Africa and potentially could serve as the source of long-distance dispersal reaching the island. However, these studies are really limited by the availability of taxonomic and geographic sampling, particularly in West Africa and perhaps unsurprisingly throughout the Congo Basin, just for reasons of access and political stability. And so taking an approach that tries to fill in some of these gaps should provide us with more insight on the frequency and the timing and the origin of these potential dispersal events and provide a better understanding of the history of these really enigmatic faunas. So among the island's endemic vertebrates, one of the only groups that is thought to have diversified within a single island and also dispersed between islands in the archipelago are the reedfrogs. The Sao Tome giant reed frog, which, if any of you have been talking to Dan about reed frogs, you know they're not very big, they're about this big. So the giant one's about this big. But the Sao Tome giant reed frog is only found on Sao Tome Island above about 1,000 meters elevation in forest habitats. And its sister taxon, Hyperoles mulleri, which doesn't have a common name, is broadly distributed on both islands, and it occurs up to about 1,400 meters on Sao Tome and up to the summit on Principe, which only reaches about 1,000 meters. So this group provides an opportunity to jointly investigate potential colonization routes from the mainland, processes driving in situ diversification within Sao Tome, as well as dispersal between the islands and the demographic consequences of dispersal between islands. And so I chose to do this. So to investigate how dispersal and in situ diversification are driving patterns of diversification in the system, I had 3 main goals. The first was to investigate potential colonization routes from the continent. And the second was to investigate the context of speciation within a single island, and then also characterize the frequency and demographic consequences of dispersal between the islands. And so I combined multi-locus phylogeography and also a genome-wide SNP data set to address these questions. So first, we're going to investigate potential colonization routes from the continent. Fortunately for me, right when I was getting started with this project, a study identified Hyperoles ventris, which is this widespread mainland taxon, as being closely related to the island endemics, because there are over 100 species in the genus Hyperoles, and so I wasn't really sure if I was going to be able to find the most closely related mainland species, but at least here I had a starting point. So this broadly distributed species, which is actually a species complex, but it's only distributed in Central Africa, not in West Africa. And so this a priori rules out the Niger River as a potential colonization route to the islands. But either the Congo or the Ogooué rivers could potentially serve as a source for these types of vegetation rocks. So I took a phylogenetic approach and I collected samples from throughout the species complex distribution on the continent to try to differentiate between these potential dispersal routes. So here's the mitochondrial phylogeny. We have the island species are shown here in pink, and then these localities on the mainland are colored according to these clades that we find. And so you'll see that this clade A, or this green clade, is most closely related to the island endemics, and its distribution corresponds primarily to the Ogui River basin, but it also reaches sort of the western extent of the Congo River. But importantly, it does not include these samples all the way here out in East Africa. And so this indicates that either the Ogui River or maybe a western drainage of the Congo River is the most likely source for one of these vegetation rats. A species tree analysis that includes multiple nuclear genes supports this same pattern. And even though this result may not seem that surprising, that the river that is closest to the island is the one that is the most likely source, this was the first study to find this pattern. And so I think this really highlights that including range-wide sampling of these poorly known species on the continent, first of all, is going to recover a lot of cryptic genetic diversity that we do or don't know about. But also that if we apply these types of studies to the other island endemic species, that we can really start to get some big picture answers to how these species colonize the islands and from where, and whether they share a similar evolutionary history or not. So the pattern of mitochondrial divergence that we recovered in this study, which the nuclear markers that I used weren't really variable for resolving relationships among the island species, but the mitochondrial pattern suggests that dispersal proceeded from that first Sinomami of interest, the mainland species, colonized Sao Tome Island. That diversification proceeded in situ on Sao Tome, and then that Hyperolus molari colonized Principe from Sao Tome. And this is in contrast to the pattern that we observe in most archipelagos, where dispersal proceeds from older islands to younger islands, and this is called the progression rule. So obviously there are a number of limitations to using mitochondrial markers to infer these types of relationships, and so I wanted to see if this pattern was supported by a larger nuclear data set. And so I used the double digest rat-seq protocol to collect some genome-wide SNPs, including a few samples from the green clade on the continent of Sinomami of interest that was most closely related, and individuals of Amensis and then molari from the two islands. And here, this is just a small subset of SNPs because they had to be biallelic and present in this outgroup taxon. But again, we see that the same pattern of relatedness among these different lineages is supported by the nuclear data set. And so this really supports that dispersal proceeded from the younger island to the older island. And for organisms that are relying on passive dispersal via vegetation rafts to colonize islands, then it makes sense that something other than island age could be driving this pattern. In particular, the pattern of ocean currents is probably going to be more important for these types of organisms than something like island age. So unfortunately, there aren't very many studies that look at the relationships of reptiles and amphibians in this particular archipelago, but the two that do exist are consistent with a south to north dispersal progression in the Gulf of Guinea, and then this does proceed from younger to older islands and also follows ocean currents. So this same species tree analysis confirms that diversification between Amensis and molari proceeded in situ on the island of Sao Tome. And so I was interested in the evolutionary history of these lineages and how they diversified within a single island. So as I mentioned, the Sao Tome giant reed frog is only found in forested habitats above 1,000 meters elevation, and the sister taxon, Hyperolus molari, is found from sea level up to about 1,400 meters. And we find some individuals that exhibit intermediate phenotypes at elevations where the two species are sympatric. And so this suggests that there's some level of gene flow between the two species, either throughout their evolutionary history or more recently as a consequence of secondary contact. So I collected samples along an elevational transect that is a little messy because the topography on Sao Tome is very complicated, but it generally proceeds from 100 meters to 1,400 meters elevation, and then I characterize these individuals using the double digest RADseq protocol. So a number of genome-wide SNPs, and then I also collected mitochondrial sequence data. So for the mitochondrial results, I recovered two major haplotype groups that largely correspond to Hyperoleus thamensis in pink and Hyperoleus molari in yellow. And then I'm going to change these dots to squares if they're thamensis localities, and they'll remain circles if they're molari localities. So I'm going to show you the mitochondrial results on the map. And what you'll see is, so I only had 20 individuals with thamensis from 2 sites, but they always carry this thamensis pink haplotype, whereas Hyperoleus molari from sites where they're sympatric with Hyperoleus thamensis, a number of those individuals carry the thamensis mitochondrial haplotype. And so this does indicate that there's some amount of gene flow between the 2 species, which isn't very surprising. I also recovered 2 differentiated genetic groups in the SNP dataset using a Bayesian assignment method. So here we're looking at a structure plot of approximately 4,000 SNPs. And here these are the thamensis individuals, and this is also a thamensis. And then everything else is molari, and we're proceeding from high elevation to sea level. And so again, here you can see that there's very high assignment to the thamensis gene for all the thamensis samples, whereas for molari and the sites that are sympatric with thamensis, we find really high levels of admixture. And the pattern of genetic admixture can arise for a number of reasons. So it could be due to the retention of ancestral polymorphism or incomplete lineage sorting. But finding this geographic pattern where admixture decreases with increasing distance from the zone of sympatry is a lot more consistent with divergence in allopatry and secondary contact. Otherwise, we would expect to find admixture sort of randomly distributed across the range of Hyperoleus molari. So I think this is a lot more consistent with a period of allopatry and then secondary contact between these previously isolated lineages. So to quantify the extent of hybridization between these lineages, we used the program New Hybrids. And here we're again using a smaller subset of SNPs because I filtered the data to only include SNPs with a minor allele frequency above 0.2. And here, this program assigns individuals to different genotype classes. So parentals for each one, molari backcross, and thamensis backcross. And at F2 and F1, I didn't get any F1 assignments, so it's not included in the legend here. So, again, we find really high assignment, and this kind of looks like a structure plot, but it's not the same. So each individual here, this is the probability of assignment to one of these different genotype classes. So this means full assignment to the thamensis parental, and then this individual has basically full assignment to F2 hybrid. So we really, really high power to differentiate between these different genotype classes in most cases. So again, we have very high assignment to the parental types for thamensis and molari, and then in the sites where the species are sympatric, we have really high assignment to one of these different hybrid classifications. So, let me see. Oh yes. So this pattern again suggests really high levels of hybridization where the 2 species distributions are overlapping, but this drops off really precipitously once we're in allopatric parts of the species range, particularly for molari, because my sampling of thamensis is more limited. And this could indicate a number of things. So I think the 2 main possibilities are that this is recent contact between the 2 species, and so we're only seeing hybridization where the 2 species physically overlap because they haven't had very much opportunity to hybridize, or that there's very strong selection against hybridization, and that's why we don't see hybridization proceeding through the rest of the molari distribution. So I am a little bit reluctant to think that there's very strong selection against hybridization, primarily because these are all adult individuals that we collected, and they're all sexually mature and breeding. But I wouldn't rule it out completely because they still could suffer some other selective disadvantage just in terms of ability to attract mates or survival based on some other traits that we don't really know much about. But they certainly seem to be viable, and the presence of back-cross individuals certainly supports that. So if hybridization is really common in sympatry, then how did these species diverge in the first place? And one potential mechanism for allopatric divergence on this island is a difference between species in terms of the types of breeding sites that they seek out. So Hyperoleus molari, which is the broadly distributed species found at low elevations up to about 1,400 meters, breeds in slow-moving streams and water-filled ditches and pig troughs and basically any standing water, they'll use it. And this is really typical of breeding habitat for reed frogs on the continent as well, including the mainland sister taxon, Hyperoleus and amamioventris. And these types of habitats are readily available across Sao Tome, but as you can imagine, once you get to higher elevations, there's really not much standing water up here. There's no streams, and certainly no water is collecting in ditches. But there is water in tree hole cavities. And this is where Hyperoleus thamensis, the higher elevation large body species, breeds. So potentially, this geographic segregation and the types of breeding habitats that are available on Sao Tome could have led to allopatric divergence between these species, and also the evolution of this really specialized reproductive mode in Hyperoleus thamensis. And this same mechanism could explain what's brought these previously isolated species back into contact. So here I am standing in the middle of the hybrid zone, and this is an agricultural field. And the main sites that we find, or actually the only sites that we're collecting these individuals in, are associated with agriculture. So cisterns or water-filled tarps that are collecting water in places where normally there wouldn't be any standing water available. And this is where we're collecting all these hybrid individuals. So, and I want to say, so this is also the same types of elevations and habitats that the really well-studied Drosophila hybrid zone between Drosophila yucuba and Sao Tome occur. And those species are also ecologically differentiated, and it's thought that the expansion of agriculture to higher elevations is what brought those two Drosophila species back into contact. And so it's possible that the same mechanism has brought these previously isolated reed frog lineages into secondary contact at these intermediate elevations, simply by increasing the availability of breeding sites where previously they would have been absent. Although our sampling of thamensis is more limited, and I'll be perfectly honest, that's because we only know of one tree right now that they breed in. So these, 19 of these come from one tree. It's a big tree that has 2 water-filled cavities in it that we consistently find them in. And the other one I'll tell you about in a minute, the other frog. So our sampling is more limited for thamensis, but not because we haven't tried. It's a very specialized frog, and it's very hard to find. But we don't find any individuals that have the molari mitochondrial haplotype. And in contrast, we find a pretty significant proportion of Hyperoleus molari that carry the thamensis haplotype. And likewise, if we look at the frequency of these different hybrid parental genotype or hybrid genotype classifications, we find that there are many more Hyperoleus molari backcross than Hyperoleus thamensis backcross. And so this is consistent with an asymmetrical pattern of hybridization, proceeding primarily from Hyperoleus thamensis into Hyperoleus molari. And this apparent asymmetry could result from 2 main types of mechanisms. So the first would be sexual differences in dispersal and mating behavior, which has been found in some other systems. So it's possible that male Thomensis, so males of the tree hole cavity breeding species, are always calling from tree holes, and they're only going to encounter females of their own species. Whereas females of Thomensis may visit both their own species breeding habitats and then also these artificial breeding habitats where males of the other species are present. And the calls of the two species are not very different, and I've collected a female Thomensis, the large-bodied species, in Amplexus with a male in a cistern in the middle of an agricultural field at about 1,100 meters elevation. So this is the hypothesis that I prefer at this point, but my sample size is very small. But that's the only female Thomensis that we've ever found not in a tree hole cavity. And we've never found a male not in a tree hole cavity. The alternative is that there's selection against one of the crosses and not the other. So if there's something about the cross between a female molari and a male Thomensis that makes those progeny less viable than the reciprocal cross. But again, I only found this hybrid zone about a year and a half ago, so we don't know yet. But those are the two main possibilities. So finally, Hyperoleus molari, the lower elevation species, is found on both islands. And so I was curious about the frequency of inter-island dispersal and whether the populations on these two islands have begun to diverge. So again, using the same RAD-seq SNP data set and then also a mitochondrial marker, I characterized divergence for a number of populations on both islands. And the pattern is consistent across both types of markers, that the populations are very strongly differentiated, which supports that dispersal between the islands was not due to humans, so it's independent of human movement between the islands, and even still, even though it did happen, it's not ongoing, so it's probably quite rare. And to further support this, so the islands are only separated by about 100 kilometers, but none of the other endemic amphibians that occur on either of the islands have successfully dispersed to one of the other islands and colonized that island. So as far as we know, this has only happened once within the history of this, or successfully once within the history of this archipelago. Another thing that is maybe a little bit surprising, so even though Principe is 30 million years old, this dispersal event occurred sometime in the last million years, it looks like, which is really recent in this island's history. And so we tend to think of successful dispersal and recruitment on older islands being determined by the availability of ecological niches. And Principe does host two other amphibian species, a large-bodied tree frog, and this time it really is a giant tree frog. It's one of the largest tree frogs in Africa. It's like this big. And then a puddle frog, which is just like a leaf litter species. And so it's unlikely that these two species would be competitively excluding something like Hyperoleus molari. They have pretty different niches. So even though we tend to think of in situ diversification as eclipsing dispersal in islands that are older, for species that don't disperse very easily overseas and that remain relatively depauperate on islands, dispersal could actually continue to play a really important role in generating diversity. So to summarize this part of the talk, I found that there are multiple possible colonization routes for organisms that are dispersing from the continent to this archipelago, and that characterizing cryptic genetic diversity in these widely distributed mainland species is really important for understanding the history of these island endemics. For in situ diversification on Sao Tome, I found support for a model of allopatric divergence with more recent secondary contact, potentially facilitated by the expansion of agriculture to intermediate elevations to facilitate sympactory between the species. And finally, I found that even though dispersal between Sao Tome and Principe is apparently possible for amphibians, it's very uncommon. And I don't know how many of you know Bob Drews at the California Academy of Sciences, but he keeps asking me if they're different species. I think because he has a really invested interest in these islands, and he really wants them to be different species? And the answer is, I don't know, but that's something that I'm hoping to work on while I'm here. So I'll be spending a lot of time at the Calicut measuring frog toes, I guess. Looking for some kind of phenotypic difference, even though it does appear that there's no gene flow between these populations, unsurprisingly, and that they probably are on separate evolutionary trajectories at this point. So I'm going to give a very brief introduction to what I'll be working on here, because it's pretty different from what I've been working on, or what I have been working on at Cornell, even though I am planning to continue some of those projects. But what I'm planning to do over the next 2 years at the MVZ is understand the evolution and function of sexual dichromatism in African reed frogs. And sexual dichromatism is just a form of sexual dimorphism where males and females are different colors. And I think you'll see soon why I am really interested in this. So there are two main classes of sexual dichromatism, and I make a distinction between the two because I think they probably differ in terms of the underlying genetic mechanisms, but also their function. So the first is dynamic sexual dichromatism, and this is a temporary color change that occurs in males during the breeding season. And the second class is ontogenetic dichromatism, and this is a case where either males or females undergo a permanent color change at the onset of sexual maturity, and this color change is maintained throughout the rest of their lives. Currently, ontogenetic dichromatism is documented in about 100 species of frogs, and obviously you can't read any of this, but you can see that there's colors at different parts of the tree, and that's just to highlight that even though this trait currently is not super common in terms of the percentage of total frog species that are dichromatic, it is found across the inherent phylogeny. So it is something that we see popping up over and over again across frogs. But the African frog family, Hyperoliidae, contains over 30% of the world's ontogenetically sexually dichromatic species. So here we're looking at just a very small subset of the phylogeny. And for those of you who know what Dan Kornick's project is, this is not his super tree. This is a very, very small subset of that. But you can see even at this level, so here are the species that have ontogenetic sexual dichromatism are highlighted in orange. And even at this level, without the full 200 species represented, there are multiple transitions from monochromatic to dichromatic states. And so that makes this group a really strong comparative study system, basically, for looking at the evolution of this trait and also the function of this trait. And among these species, there are a number in which females appear to have more conspicuous coloration relative to males. So this is MVZ, so I imagine everyone knows, but the female is the bigger one, the male is the smaller one. And then there are also species in which males and females are just different colors, but it's not really clear that one of them is maybe more cryptically or conspicuously colored than the other. And this is Hyperolius cinnamomia vectris, which is the mainland sister taxon to the island endemics. So I think that's all. Oh yeah, and so seeing these multiple evolutionary transitions throughout the Hyperoliidae phylogeny suggests that this trait is evolutionarily labile. And that makes sense in terms of what we think is the underlying mechanism for how these ontogenetic color changes are mediated. So a lot of these species have a distinct juvenile and adult coloration, and we think that steroid hormones at the onset of sexual maturation are what triggers this change in dorsal coloration. And so in some species, both sexes undergo a color change, and so they remain sexually monochromatic as adults, but they don't retain the juvenile coloration. And if the color change pathway is disrupted in one sex, and in this case it's the male, then only the female would undergo the color change, the male would retain the juvenile coloration, and now we have sexual dichromatism in this species. So as I mentioned, in many species, the females have what appears to be a bright coloration compared to males. And we can imagine, based on what we know from studies in other species that have female-biased dichromatism, it's called, or brightly colored females, that this could play a role in mutual mate choice or female-female competition for some sort of limited resource. But there's also the possibility that other factors that play throughout the rest of the year, like differences in predation pressure, and that differences in crypsis, depending on the type of habitat that you're spending your time in, could be driving some of these differences between the sexes in coloration. And so this hasn't been investigated in any amphibian as far as I know, but there is some evidence that male reed frogs, male hyperoleans, experience greater predation pressure when they're calling at breeding sites relative to females. And so this is something that I'm hoping to address in my time here at the MDC. So to try to understand the genetic basis of this trait and also the function of this trait, I've chosen to focus on a species that is polymorphic with respect to sexual dichromatism, and it happens to be found throughout Central Africa, but also the land-bridge island Bioko, which became isolated from the continent most recently about 12,000 years ago. And the main reason that I'm focusing on this population, other than the fact that it's polymorphic, which will make sense in a second, but also because it's an island, there are no really large dangerous mammals around, which is very important for spending time in the field and trying to observe the behavior of your focal species. So there are no elephants there, and there are no forest buffalo and gorillas and those kinds of things. So this species, Hyperoleus tuberculatus, which also does not have a common name, is found only on the southern parts of the island, the only place that I found it there. And it breeds in sort of swampy forest clearings for the most part. And neither sex, as far as we know, provides any type of parental care. And I guess I should have mentioned, as far as we know, none of the hyperoleids are chemically defended. So we don't think that this coloration serves, in that sense, as communication to potential predators. So most of the males in this population retain the juvenile coloration, and the females always undergo an ontogenetic color change, and they become this kind of reddish, it depends, it varies, but usually reddish coloration. And a small subset of the males also undergo the color change. And so this means that I can compare individuals that have the same coloration phenotype, but also different sexes, and then vice versa. So the plan over the next couple of years is to try to link gene expression in males and females to skin pigment composition, to quantify changes in gene expression when males are induced to have the ontogenetic color change. So if I expose males to estradiol for long enough, they will assume the female coloration. So we can characterize gene expression changes that happen when we induce the color change. And then finally, to characterize coloration and crypsis and habitat use between the sexes and the different color morphs in the field. And so we'll see how much of that I actually get done, but that is the plan for my time here. And so with that, there are a lot of people that I would like to thank for help in the field and for funding to get out to Africa all the time. And I'm assuming I'm not the only person to have done this, but my dissertation was actually dedicated to the MVZ, and that's because the reason that I got really excited about science and that I've continued on this track is entirely due to my time spent here as an undergraduate, and I'm very grateful for that, and it's really, really nice to be back here. And I was told that there was supposed to be a poem at the beginning of this talk. I don't know if that's Michael.
I'd be happy to read it.
Well, it's okay because I have another poem. So I wrote some blog posts for National Geographic when I was on an expedition to Sao Tome and Principe, and this is a poem that someone wrote in response to one of my blog posts, and it's one of my favorite things. questions. Thank you.